Hello everyone. In this lecture today I'm going to talk to you about nucleases. I'm going to talk to you about this nucleases, but my focus of today's talk will be on restriction endonucleases, or simply restriction enzymes. Okay, so first, what are nucleases? Okay, these nucleases, they are the enzymes that degrade nucleic acids. Okay, so basically nucleases, as the name suggests, they are the enzymes that degrade nucleic acids. So basically this is the opposite function of what polymerases do. Okay, polymerases are the another categories of enzymes. So then how these nucleases, they degrade nucleic acids. Nucleic acids, it can be DNA or it can be RNA, right? So these are the nucleic acids. So what these nucleases do, they degrade it. So how do they do that? They hydrolyze or break an ester bond in a phosphodiester linkage. Okay, so these nucleases, they break an ester bond in a phosphodiester linkage between adjacent nucleotides in a polynucleotide. For example, here in the B here, this linkage is phosphodiester, because on this side we have one ester and the other on the other side also. So this B indicates the phosphodiester linkage. So these nucleases, they actually hydrolyze or break an ester bond. Okay, so an ester bond in a phosphodiester linkage that is present between the adjacent nucleotides. Okay, so between adjacent nucleotides. So then, and the next thing is, okay, so we know what are nucleases. So nucleases, they have been divided into two types. One is endonucleases and the other is exonucleases. As the name suggests, endonucleases, so what they do, these endonucleases, they hydrolyze internal bonds, okay, within the polynucleotide chains. So endonucleases hydrolyze internal bonds, whereas exonucleases, okay, exonucleases, they start at the end of the chain and hydrolyze from that end position. Okay, so basically if we go back to our previous slide, so here guys, the endonucleases, for example, they hydrolyze internal bond. Okay, it's shown here, this C, they hydrolyze these internal bonds, whereas the exonucleases, they can act from, okay, this side, so from the end, and what they do, they start at the end of the chain and hydrolyze from that end position. So these are the exonucleases. So this is the difference. This is the difference between these two different types of enzymes, okay, nucleases. The specificity of nucleases, it ranges from non to extreme. That means that nucleases may be specific for DNA as DNases, or for RNA as RNases, or even specific for both DNA and RNA hybrid. For example, RNase H, which cleaves RNA strand of hybrid duplex. Okay, so this is point number one. And the another important point is that restriction endonucleases, they are also called restriction enzymes. They are from eubacteria and archaea that recognize specific DNA sequence. Okay, so restriction endonucleases are from eubacteria and archaea, and they recognize a specific DNA sequence. The function of restriction endonucleases is mainly a protection against foreign genetic material, especially against bacteriophage DNA in these prokaryotic organisms. So naturally, the function of restriction endonucleases, or simply restriction enzymes, is mainly protection against the foreign genetic material, especially against the bacteriophage DNA. Okay. So the synopsis of this slide is, guys, the nucleases, they are of two types, endonucleases and exonucleases. So then further, the nucleases, they have been divided into four different types, like type 1, type 2, type 3 and type 4. These are endonucleases. They have been divided into four different types. And so and there are some differences, but the most commonly used restriction endonucleases or restriction enzymes are type 2 type. So what is the main feature of these type 2 restriction endonucleases is that they specifically recognize sequence. The cleavage site is within or close to the recognition sequence. They generate 5' phosphate and 3' hydroxyl termini at cleavage site. And these enzymes, this category of enzymes, they require magnesium. Okay. when it comes to type 1, guys, these type 1 enzymes, multi-subunit protein, both restriction and methylation activities, they also require ATP, and cleavage site is at a variable distance from the recognition site. Okay, so cleavage site is at a variable distance from the recognition site. But here for type 2, the cleavage site is within or close to the recognition sequence. For type 3 restriction enzymes, 2-part recognition sequence in an inverse orientation, and cleavage sites are at a specific distance away from one of the recognition sequence. And for this class also there is a requirement of ATP. And type 4 restriction enzymes, the cleavage of only methylated DNA. So these type 4 restriction enzymes, they only cleave methylated DNA. This is a really important point, guys, that you should remember. And the cleavage site is approximately 30 base pairs away from the recognition site. Okay. So out of these different types of restriction into endonucleases, as I said earlier, the most commonly used restriction endonucleases are type 2 restriction endonucleases, or simply restriction enzymes. And now moving to the next point, guys. So then how these restriction enzymes, or simply restriction endonucleases, how they function. Okay, so let's take an example of one restriction enzyme called EcoR1. Okay, these restriction enzyme, they recognize a site, a sequence in a DNA. That sequence is called palindromic sequence. Okay, so this sequence is palindromic in nature. This is not called palindromic in nature, but this is called the recognition sequence. Okay, so this is the they recognize this sequence. So basically, for example, we took the example of EcoR1. This is a restriction enzyme. It recognizes what? The recognition sequence in a DNA. So this recognition sequence is around 4 to 8 bases, and this recognition sequence is palindromic in nature. So what does it mean, guys? So palindromic means, if you read from this side, so GAATTC, and if you in this strand, if you read from this side also GAATTC. So basically we can read the same from one this direction, and the other from back and front. So we read the same thing. So this kind of sequence is called palindromic sequence. GAATTC here from this side, also GAATTC. So this kind of sequence is called palindromic sequence. So this palindromic sequence is recognized by our restriction enzymes. Okay. So then each restriction enzyme, they have their specific recognition sequence. So this is the recognition sequence for EcoR1. And so they recognize this sequence, and they will cleave this EcoR1. It will cleave this from this side, the side from GA here. Okay, this is where this restriction enzyme will cleave on this strand, whereas from here it will cleave on the other strand. So then what will happen, guys? Here G will be left, right? Here also we will have this. So here we will have this sequence left, and here we will have this sequence left. So what kind of this kind of restriction digestion, or this kind of cleavage, this leads to the 5' and 3' overhangs. Okay, so this is 5' overhangs, you can see here 5' overhangs, and here this is 3' overhang. Okay, so this 5' and 3' overhangs. So this is called staggered cut, and it generates in sticky ends. Okay, sticky ends. Okay, so these ends are called sticky ends. Okay, so once again, guys, EcoR1, it is a restriction enzyme. It recognizes a specific sequence. This sequence is called recognition sequence, and this recognition sequence is palindromic in nature. That means that we can read the same from backward and the front, GAATTC from in this direction if we read it, and GAATTC in this direction if we read it, right? And then this is the recognition sequence. And where does it cut? Okay, whenever this enzyme sees this sequence, it will recognize it, and it will cut from here and here, and then it will result in these overhangs, 5' and 3' overhangs. And this kind of restriction digestion is called staggered cut that results in sticky ends. Okay, so with these sticky ends. So if we take the another example, guys, here of this restriction enzyme SAE third. So the recognition sequence is this. So this is GGCC, CCGG. If we read from this direction, GGCC, and if we read from this direction, GGCC. So this is also palindromic in nature. Yes, this is palindromic in nature. And another important point is this is the recognition sequence. This is palindromic in nature, and it will cut in the middle, guys, from here. And therefore, we will have 2 bases here, 2 bases here, 2 here. So here the ends are not sticky, okay? Here the ends are sticky, as you can see, 5' and 3' overhangs, but here the ends are not sticky. This kind of cuts, because it's cutting from directly separating, you know, like two bases apart. So this kind of cut by the restriction enzyme is the blunt. Okay, it results in the blunt cut. Okay, so it's a blunt ended. The sticky ends here we got sticky ends, but here we get blunt ending, guys. So this is the difference. Okay, the sticky ends and the blunt end. Okay, so this is the common way how the restriction enzyme function. They recognize the recognition sequence and they cut it. One type, for example, EcoR1, it results in sticky ends. For example, SAE1, it results in blunt ends, okay, without 5' and 3' overhangs for SAE third. And the next important thing is, guys, even though I did not mention, so how these restriction enzymes are named, okay, so what is the system of the nomenclature. So let's take an example of EcoR1, guys, okay, so ECOR, R and then 1, okay, so I did not write it correctly, maybe I should make it italicized, eco, and then R and then 1, okay. So if we look at this enzyme, you see these first 3 letters, they are italicized, and this first E of this restriction enzyme, if an enzyme has a name EcoR1, this first letter E is the name of the genus of the bacteria. That means Escherichia coli. Because this EcoR1, it comes from the Escherichia coli, so this first letter is this E from Escherichia coli. Okay, and CO, okay, so and then these second and third letter, the CO, these 2 letters are from the species, okay, species of the same bacteria. That means CO, okay, you see ECO, so ECO, and these 3 letters are italicized. And this fourth letter R, it refers to the strain from which this enzyme was obtained. So for this EcoR1, it was obtained from RY13, therefore we have R here. And this Roman numeral 1, okay, so this Roman numeral 1, it indicates, as you can see, indicates the chronology of the identification. Basically, it is the first enzyme. 1 indicates it is the first enzyme that has been isolated, that has been obtained from this Escherichia coli strain RY13. Okay, so basically the nomenclature, you know, like I explained, the first 3 letters, they are italicized, and the first letter of the enzyme is the first letter of the genus of the bacteria from which the enzyme was isolated. And the next 2 letters are the 2 letters of the species of the same of the bacteria from which the enzymes are isolated, and this is followed by extra letters or numbers to indicate the serotype, and a space. Sorry, I forgot the space, and in between eco and space, and then Roman numeral. Okay, so then third letter. So after these 3 letters, R, it refers to the strain, and then we have a space and the Roman numeral to indicate the chronology of identification. Like I said earlier, EcoR1, it indicates the first endonuclease which was isolated from Escherichia coli strain RY13. Therefore, it is named as EcoR1. Okay, so this is the nomenclature, guys. Now moving to the another really important point. The important point is that you know, there are type 1 and type 3 enzymes, this restriction endonucleases, they differ, like I said earlier, they differ from type 2 enzymes in the recognition site, and the cleavage sites are different and usually not palindromes. Okay, so for these type 1 and type 3, okay, this is one point that you can just make a note of. Okay, but what is the most important thing that I want to explain here is isoschizomers. Okay, iso means same, guys. As you know, iso means same, and schizo is like scissors. Okay, schizo is like scissors. So what are isoschizomers? What are isoschizomers? Isoschizomers are that scissors means cutting. Okay, S-C-S-S-O-R-S, scissors. Iso means same, and schizo means scissors. Okay, so iso means same and schizo means scissors. Basically, isoschizomers are two enzymes, okay, that have the same recognition sequence and cut in the same location. Okay, they have the same recognition sequence and cut in the same location. Okay, these are isoschizomers. Okay, iso means same and schizo means, for example, schizo means scissors. For example, SPS1 and BBU1. Just look at these guys. So for SPS1, the recognition site is GCATGC. This is the recognition site for SPS1. Whereas the recognition site for BBU1 is also GCATGC, right? This is the recognition site for BBU1. They have the same recognition site, and they cut in the same location. This enzyme cuts here and here, as you can see, SPS1, and also BBU1 also cuts here and here. Therefore, these two, SPS1 and BBU1, they are called isoschizomers because they have same recognition site, same recognition sequence, and they cut in the same location. Okay. Another really important point is neoschizomers. Okay, guys, neoschizomers. Okay, neoschizomers. So what are these neoschizomers then? Neoschizomers, they recognize the same nucleotide sequence, but they cleave at a different position. Okay, so the difference between isoschizomers and neoschizomers is that the neoschizomers and neoschizomers are the restriction enzymes. They recognize the same nucleotide sequence. So basically, two restriction enzymes. This is one restriction enzyme. This is another restriction enzyme, SMA1 and XMA1. So this SMA1, they have same recognition sequence. If you look at this, the recognition sequence is CCC and ZZZ, and it's also the recognition sequence for XMA1 is also CCC and ZZZ. But if you look carefully, they are cutting at the different location. So SMA1 is cutting here, and it is generating blunt ends. Cutting here, and it is generating blunt ends. Whereas the XMA1 is cutting here and then here, right? So it is generating sticky ends. So they have the same recognition sequence, SMA1 and XMA1, but different. But they cut at the different location. These kind of restriction enzymes are called neoschizomers. Okay, neoschizomers. Okay, and another important point is what are the different factors that affect the restriction enzyme activity. Okay, so the factors are temperature, cofactors, ionic conditions, buffer systems, and methylation status of the DNA. Okay, so temperature is most endonucleases, most restriction enzymes. They digest the target DNA at 37 degrees Celsius with few exceptions. Some enzymes, they work better at lower temperatures. For example, 25 degrees Celsius. Example is SMA1, while TAC1, another restriction enzyme that works at 65 degrees Celsius, let's say. So the temperatures is one factor that affects enzyme activity. Another important factor is the cofactors. Restriction enzymes require certain cofactors or combination of cofactors to digest at the recognition site. For example, all restriction enzymes, they require magnesium as a cofactor for the endonuclease activity. Okay, and third important factor is ionic conditions. Magnesium is required. Magnesium is required for all endonucleases, but some enzymes also require ions such as sodium and potassium. Buffer systems. Most restriction enzymes are active in pH range 7 to 8 pH range, and treats HCl as a buffer, which is a temperature dependent buffer that is most commonly used. And final, but the really important point is the methylation status. Methylation status of the DNA. Methylation of adenine or cytidine residues affects the digestion of DNA. Okay, so it's also really important. I will discuss in detail about the methylation of the DNA and how it affects the restriction enzyme activity. So methylation status of DNA strand, it is one of the most common types of DNA modification that is found in eukaryotes and prokaryotes. In bacteria, it is a part of the restriction modification defense system against the bacteriophage infection, whereby host bacteria, they protect their own DNA against cleavage by endogenous restriction enzymes. Methylation commonly occurs at the cysteine residues and also at adenine residues, but predominantly forms 5-methyl cytosine or 4-methyl cytosine or 6-methyl adenine, okay, derivative. So it's basically 5-methyl, 4-methyl cytosine and 6-methyl adenine derivative. So these are the places, these are the sites where methylation occurs. Okay, the nucleotides bases where methylation occurs. DAM, it stands for deoxyadenosine methylase transferase, and it converts this sequence into this methylated sequence, whereas DCM, it stands for deoxycytosine methyl transferase and converts the sequence CCWZZ to this WZZ3, where W is either A or T. So then, okay, so this is all the theory I said. Okay, I explained. So basically methylation, it commonly occurs. Methylation is the addition of the methyl group, where this methyl group is added. The methyl group is added at the cytosine or the adenine base, and normally the derivatives are 5-methyl cytosine, 4-methyl cytosine and 6-methyl adenine derivatives. And so how then it affects the restriction enzyme activity, that I'm going to discuss here. For example, this is here unmethylated DNA. When it is unmethylated DNA, then this MBO1, okay, so when it is unmethylated DNA, MBO1, it can cut this restriction site. Okay, this restriction site, MBO1 it can cut, but DPNL cannot cut. But when it is methylated at the 6-methyl adenine, this site, so adenine site, then what will happen is that this MBO restriction enzyme, its activity is blocked. Okay, it cannot cut. But this another restriction enzyme, DPNL, it can cut. Okay, so this is one example. So that means that methylation of the DNA, it can block the restriction digestion by the enzyme. Okay, this is one point. It can not only block, guys, but another thing is it can results in partial digestion. For example, for XHO1, okay, so for XHO1, when there is no methylation, so it can easily cut, recognized in this sequence. But when there is methylation at this cytosine, 5-methyl cytosine, then what happens is that the cut by the XHO1 is it's the restriction. There is partial restriction digestion by this enzyme. So the synopsis of this slide is, guys, by giving all these examples, what I want to mention to you is that the methylation of DNA results in either complete blockage of the restriction digestion, or it can even lead to partial restriction digestion, or it may not affect, anything. Any of these three possibilities can happen. I hope, guys, this video was helpful. Thank you very much for your kind attention. Please subscribe our channel to support us. Thank you very much.